Good afternoon, everyone. Thank you for attending the next Real World Evidence Forum. I'm Dr. Pablo Salcedo. And in this session, we're going to be discussing the test case titled Real World Clinical Outcomes in Patients with Mechanical Heart Valve Replacement and Anticoagulation Variability. I'll start by introducing some key personnel for this test case. We have Dr. Julie Prillinger. She's a principal scientist for the Global Data Science and Analytics Division at Abbott. We have Dr. Pingsheng Wu. She's the Research Associate Professor for the Division of Allergy, Pulmonary, and Critical Care Medicine at Vanderbilt University Medical Center. And we also have Rebekah Ward, epidemiologist from the Heart Valve Devices team and the Office of Cardiovascular Devices. So some background facts about this test case. The data sources were electronic health records and registries retrospectively. The duration of the study was about 14 months. It completed this year in January. The disease area is cardiology. The technology of interest is a mechanical aortic heart valve. The overall aims of this study included trying to test the feasibility of identifying patients who received the mechanical aortic heart valve, as well as studying longitudinal clinical outcomes in patients with varying INRs. So when looking at the test case relevance and the criteria that was used in selecting this test case on behalf of NEST, this test case assesses the feasibility of linking various sources of existing real-world data to study longitudinal patient outcomes. In addition, this project intended to develop query algorithms that in the future could be shared with NEST and its collaborators to guide future studies and develop a national cohort of patient recipients. So the importance of this device in the medical device ecosystem, there's a high clinical burden and disease burden for heart valve diseases and other valve-related diseases. Valvular heart disease affects tens of millions of patients globally and has a high rate of morbidity and mortality. Treatment of severe aortic valve disease often includes surgical replacement with a prosthetic heart valve, and this can add years to a patient's lifetime. Insights drawn from this test case have the potential to impact long-term clinical care outcomes for patients who have received a mechanical heart valve. A little bit about NEST's role in this test case. It was twofold. One was guiding the test case administratively as well as technically by check-ins, preliminary reports, and monthly progress reports. This allowed NEST to keep a close eye on timeline and deliverables and to ensure nothing fell off track schedule-wise. In addition, through the assignment of a research scientist by NEST to the test case, additional technical expertise regarding data quality as well as ensuring compliance with the NEST research methods and data quality frameworks was ensured. Now I hand it over to Julie from Abbott.
Great, thank you so much. We can go to the next slide. Great, well, it's really a pleasure to be speaking here today at the Real World Evidence Forum and to be sharing our experience in a NEST test case. I wanted to start today by just providing some clinical context and to take a minute to share why, as a submitting organization, Abbott was interested in pursuing this scientific question. So Pablo touched on this a little bit at the start, but valve disease impacts over 40 million people globally. And then in the US, it's estimated that about 10 to 15% of those over age 75 are affected. Aortic stenosis is a common form of valvular heart disease, and valve replacement surgery is indicated when this manifests as patient symptoms. So this can include fatigue, shortness of breath, chest pain, or when there's evidence of significant valvular impairment. Either mechanical or biological valve prostheses can be used. And for mechanical heart valves, which were the subject of this test case, these are made of durable materials, usually titanium or carbon, which can potentially last a patient's lifetime, but do require lifelong oral anticoagulation to protect against the risk of thromboembolism. So the anticoagulant currently used in this population is warfarin, and that's managed by actively monitoring a patient's INR levels, which is a measure of blood clotting time. In general, higher INR levels are associated with greater risk of bleeding, while low INR levels may be associated with thromboembolic risk. Several medical societies have published guidelines for anticoagulation in patients with mechanical heart valves, and the 2014 AHA-ACC guidelines recommended maintaining an INR range between 2.0 and 3.0 for patients implanted with a mechanical heart valve. However, there is some data to suggest that a reduced INR regimen may be beneficial. And this is really important because the percentage of clinically relevant bleeding events can be 3 to 4-fold higher than the percentage of thromboembolic events following a mechanical heart valve implant. So our goal in this NEST test case was to evaluate whether we could use real-world data to study clinical outcomes associated with a reduced INR regimen following aortic valve replacement with an Abbott mechanical heart valve. So this was intended to be a retrospective study, and it really was predicated upon the concept illustrated here. So that is, while the majority of patients implanted with Abbott valves likely have INR measurements in the guideline recommended range of 2.0 to 3.0, there are likely also populations of existing patients who have INR measurements either above or below that range. And so if we're able to identify those patients and study their longitudinal clinical outcomes, it may provide guidance on optimal medical management for all patients implanted with mechanical heart valves. Next slide. So at the time of submitting our test case proposal, Abbott was really interested in partnering with the NEST network collaborators to study this clinical question. And there were several advantages to this approach. So first, this represents real-world clinical management following a surgical intervention. And as Dr. Wu will show, this is particularly important for this clinical question, given the variability in INR levels that we observed for these patients. And ultimately, what we want is a data set that reflects the reality of warfarin management for these patients. Second, with many real-world data sets, we're often able to identify a large patient population that can lend greater statistical power, particularly for studying rare clinical events such as stroke or mortality, which were of interest for this study. Third, as a medical device manufacturer, we really are motivated to understand how we can use real-world data towards regulatory applications. And in this case, we were interested in understanding potential labeling expansion for a commercially approved product that's been on the market for a long time. And then finally, I think big data and machine learning and AI have really become buzzwords in healthcare, but there is the potential to eventually use some of these data and these tools to predict the optimal INR for a single patient and really move us towards that goal of truly personalized medicine. So hopefully that set the stage, and I'm now going to hand it over to Dr. Wu to walk through our study design and findings from this NEST test case.
Great. Thank you, Dr. Pringer. Again, this is a test case study. It is a collaboration between Abbott and NEST partners, Vanderbilt University Medical Center and Weill Cornell Medicine. Next, please. So as a test case, we propose to examine the feasibility of subject identification, whether we can identify subjects who have had the Abbott-specific aortic mechanical heart valve in the electronic health records. Once we identify the subjects, we propose to perform chart review to characterize the subject's population, particularly their anticoagulation management, following the aortic valve, the mechanical heart valve implantation, namely whether we can identify and determine the international normalized ratio INRs in the electronic health records. Lastly, we would like to evaluate the feasibility of studying clinical outcomes that are associated with reduced anticoagulation following Abbott aortic mechanical heart valve. Two things. First, whether we can capture clinical outcomes and what is the real-world anticoagulation management, the INRs look like in this patient population. Next, please. So the study population includes subjects who have had an Abbott aortic mechanical heart valve implantation at either Vanderbilt University or Cornell Center during 2010 to 2017. We identify subjects using registries and a computable phenotyping approach. The registries, both Abbott Registry and Vanderbilt Hospital Registry, contains information regarding subjects' name, date of birth, gender, date of implantation, and Abbott-specific model, or the specific model used. The computable phenotyping approach, showing the diagram here, is an algorithm of combination of ICD-CPT codes and anticoagulation specific medication use and lab measurements. And lastly, the algorithm also implements a natural language processing approach, searching the clinical notes to narrow down to Abbott-specific models. Next, please. So we identified a total of 248 subjects, 223 from Vanderbilt and 25 from Cornell. Since we implemented all three approaches in Vanderbilt electronic health records, and the diagram in the left shows the number of subjects identified from each of the three approaches. And for Cornell, we only applied the Abbott registry. Next, please. So our study population is further limited to patients who were at least 18 years of age at the implantation, and the implantation at and only at the aortic position and with Abbott-specific model. 186 unique subjects, 170 from Vanderbilt and 16 from Cornell were included in the final study. Next, please. So I mentioned that we applied three approaches, identify subjects at the Vanderbilt site. This slide shows the approach-specific number breakdown in getting our final study population. We can see that the hospital registry, the Vanderbilt Hospital Registry, identified the most number of subjects, 165 out of 170, followed by the Abbott Registry, 151 out of 170, and the computable phenotyping approach basically has the highest probability in missing the subjects of interest, with only 108 subjects identified. Next, please. So subjects were further classified based on the guideline recommendation for the management of patients with the aortic mechanical heart valve. Subjects who with stroke, with chronic or persistent atrial fibrillation, or with left ventricular ejection fraction less than 50% were classified or considered as having high risk, and their target INR is between 2.5 to 3.5. And subjects with no above conditions were classified or considered as having low risk, and their target IR is between 2 to 3. And the table here shows the corresponding low INR, subtherapeutic INR, therapeutic INR, and the supratherapeutic range for low risk and high risk subjects, respectively. Next, please. And we have 103 subjects classified as low risk, 83 subjects classified as high risk. And there's not much difference in age at implantation, sex, race, smoking, and study site between the high and the low risk group. The average age at implantation is about 51 years, 62% were male, and predominantly is male, is non-Hispanic white. And next, please. And compared to the low-risk subjects, high-risk subjects are more likely to have diabetes, hypertension, coronary heart disease, heart failure, and for the cardiac pathology, less likely to have aortic stenosis. And the left ventricular ejection fraction here, high risk subjects lower, that's by definition. They should be less than 50%. Next, please. So we successfully identified subjects, the INR, in our electronic health records. And notice that the sample size here is 184 because 2 subjects died at the same hospitalization of the implantation hospitalization. And the figure at the left side basically shows that the subject's INR follow-up could be divided into roughly into 3 patterns. So the plot here, the x-axis is days after implantation, so 2 years after implantation. The y-axis is the INR values. We can see that the top panel represent the 70 subjects. They have short duration INR monitoring, so less than 3 months. And the middle panel, that's the, we have 41 subjects. Their INR monitoring has at least 1 gap greater than 2 months in between. Lastly, we have this 73 subjects represented in the bottom panel. They have sufficient INR monitoring. Next, please. So this table shows how the subject's INR or anticoagulation was managed for both high and low risk group. And we can see that the median follow-up time for low risk group is about 10 months. And for high risk is about 15 months, so a little bit longer. And the median total INR measurement is 30 for low risk group and 36 for high risk subjects. And based on what we described below, the low subtherapeutic, therapeutic, and super therapeutic INR management, this the bottom 4 rows shows subjects proportion of time in each category. And for each cell, the top, that's the mean and the standard deviation. The bottom, that's the median and interquartile range. So we can see that for low risk subjects, about 60% of the time they are their time were managed in this therapeutic range. But for the high risk group subjects, only about 1/3 of the time fall in the therapeutic range, and another 1/3 of the time fall in the subtherapeutic range. So we did a chart review. We selected randomly selected 20 high risk subjects at Vanderbilt. We found that 19 out of 20 subjects, they had a physician instructed target INR 2.0 to 3.0 instead of guideline recommended 2.5 to 3.5, suggesting that the high-risk subjects may be managed similarly or per instruction to the low-risk subjects in terms of INR management. Next, please. So the last aim, we try to determine or evaluate the feasibility, whether we can capture clinical outcomes. So this is the table shows the outcomes we captured out of these 186 subjects. Five people died within the 2-year follow-up. Be aware that these 5 subjects includes the 2 people who died at the implantation hospitalization. And we identified 4 major bleeding events, 1 hemorrhagic stroke and 1 embolic stroke. And the bottom figures, that's the 4 people, or represents 4 people who have had the major bleeding and hemorrhagic stroke. Again, x-axis, that's days after the implantation, y, that's the INR value. So in each plot, we labeled the B representing the major bleeding, the time when they have the major bleeding events. So we can say what's going on between the INR values that's represented by the blue lines, the relative timing relationship. I will pass on to Dr. Pringer for the lesson learned from our test case study.
Thank you, Dr. Wu. So I think from the perspective of a feasibility study, we would really view this test case as an overall success in that we were able to build a cohort of patients implanted with Abbott mechanical heart valves at 2 sites. We were able to extract their longitudinal INR data and then identify relevant clinical outcomes. But with that, and with the nature of this being a pilot study, came a number of challenges and lessons learned, some of which Dr. Wu has highlighted already. But here we've grouped these into 3 main categories. So first, we have items pertaining to patient identification and building the study cohort. Second, we have lessons learned related to real-world INR monitoring and our now greater understanding of the variability and completeness of these data. And then finally, some thoughts on measuring clinical outcomes using EHR data. And I'll walk through each of these now. So as Dr. Wu outlined, we used multiple methodologies and data sources to identify patients who were implanted at Vanderbilt or Cornell with specific Abbott valve models. And then the team characterized metrics such as sensitivity and positive predictive value for each of these approaches. There were a lot of learnings from this exercise, and ultimately this information can inform the optimal approach for manufacturer-specific identification moving forward. So for example, we learned that in order to identify Abbott-specific heart valve models within the electronic health records, so not linking to the Abbott registration database or the surgical registry, that techniques such as NLP need to be developed. And this would in turn have implications, really, if we were to deploy across additional sites in terms of developing suitable algorithms for each valve model and hospital system. Also related to this study cohort, Dr. Wu discussed that the sample size for this test case was quite limited. It's possible that other hospitals within the NEST network may have had a larger number of Abbott valve implants, so some additional feasibility testing upfront may have led potentially to a larger patient cohort. And then we also identified significant interinstitutional differences in data availability and data format between Vanderbilt and Cornell. And it was our takeaway that further standardization of the electronic health records between data partners may be a worthwhile exercise. Moving along to the INR data, I'd say this was one of the greatest learnings from the perspective of the industry partner, because real-world INR values are data that we don't often get visibility into. We learned that missing data and inherent INR variability pose challenges for studying anticoagulation in the real-world setting. And it's possible that supplementing the hospital INR data with local anticoagulation clinics or home INR monitoring may mitigate issues with data completeness. Interestingly, as Dr. Wu pointed out, patients that may be classified as high risk and who should be managed with a higher target INR per the society guidelines appear to actually be managed within the normal therapeutic range of 2.0 to 3.0. And for us, this was an important lesson learned, both from a real-world study design perspective and when thinking about best practices in optimizing patient care. And then finally, while the team did successfully identify relevant clinical outcomes over the 2-year follow-up period, I think linking EHR to an administrative claims database or the National Death Index, for example, may help supplement outcomes for patients who are followed elsewhere or for those who have an out-of-hospital death. And finally, the research team spent a considerable amount of time reviewing and adjudicating the clinical events that were identified during this test case. But as a team, we acknowledge that the scalability of this approach may be limited. And so development of alternative approaches such as computational phenotypes may be warranted. So I think these are some of the key takeaways and lessons learned from our test case. As just one additional point of commentary, I would say that a prerequisite for success is having research collaborators who deeply understand electronic health records and their site-specific data elements. we had that expertise in our 2 network collaborators, and I think that was really vital to overcoming many of the challenges that we have listed here. Right, and so now I will pass it off to Rebekah Ward from the FDA to give some commentary.
Thank you, Julie. My name is Rebekah Ward. I'm an epidemiologist for the Heart Valve Devices team in the Office of Cardiovascular Devices and the Center for Devices and Radiological Health at the FDA. And today I will give the perspective of the Heart Valve Devices team on real-world evidence and this test case. Next slide, please. To begin, I would like to share the Center for Devices and Radiological Health mission statement. Our mission is to protect and promote public health, to assure that patients and providers have timely and continued access to safe, effective, and high-quality medical devices and safe radiation-emitting products, to provide consumers, patients, their caregivers, and providers with understandable and accessible science-based information about the products that we oversee, and finally, to facilitate medical device innovation by advancing regulatory science. Next slide, please. The FDA uses real-world evidence to monitor post-market safety and adverse events and to make regulatory decisions. The 21st Century Cures Act, which was passed in 2016, places additional focus on the use of real-world evidence to support regulatory decision-making. But it makes no change in the existing evidentiary standards applicable to these decisions. Real-world data and real-world evidence may be considered valid scientific evidence depending on the characteristics of these data. For a detailed description of FDA's perspective on real-world evidence and the use of it for regulatory decision-making, please take a look at the 2017 guidance via the link referenced here. If leveraged correctly, real-world evidence may help to reduce the time and cost for evidence generation, supporting FDA marketing authorization and post-market requirements. Next slide, please. Real-world evidence has been utilized by the Heart Valve Devices team for several purposes, including expanding indications of an FDA-approved device in the pre-market, identifying potential safety signals as part of surveillance efforts, and to address the requirements of conditions of approval for mandated studies. Next slide, please. Development of real-world evidence from real-world data is often a challenging endeavor. Test cases, including this one, are extremely valuable to our understanding of the real-world data ecosystem for medical devices. These test cases often identify potential data gaps or data quality issues that might impact the fit for purpose of real-world data for regulatory decision-making. In this specific test case, the researchers noted that there was high variability and data missingness for INR readings, difficulty in determining device type across EHRs, and that the identification of longer-term outcomes of interest will benefit from linking currently available data to insurance claims data. The researchers have also used this test case to develop strategies for bridging these data gaps, and this will benefit any future development of real-world evidence for the mechanical heart valve space. Next slide, please. The Office of Cardiovascular Devices and the Heart Valve team encourages stakeholders to engage us early and often throughout the process of developing regulatory research. This includes research utilizing real-world data. The Q submission process is a recommended means of obtaining FDA feedback on any research proposal. Please find more information via the link here. The Heart Valve Devices team thanks NEST and the research team for the opportunity to engage on this NEST test case. Thank you.